Okay.
Just let me know when you want me to start.
It is 3:29 now, so maybe give everyone another minute, and then whenever you're ready, go ahead and start.
All right, I'm going to not start before because people could rightfully complain. All right, it's 3:30 on my watch, so I'm actually going to start. I'm Chris Adami. I'm a professor in the Department of Microbiology and Molecular Genetics, and I'm happy to jump in and give this Beacon seminar. So you might think, wow, Chris, offered to do a talk really quickly. The thing is, I was actually invited to a Santa Fe Institute workshop, and I had to make a talk for them. So I figured I might as well, two flies with the same swatting instrument. So here you get to see this work, which was done here actually, not last year, but 2 years ago. But in fact, I don't think I've ever talked about this combination of topics. You also see that there's this rectangle on the upper right. I made that so that you can actually move your Zoom window where I should be appearing right there. And I made sure that nothing is behind it. So in fact, you don't have to move that thing around to discover stuff that is on my slide. So you can just leave it there and it'll never cover up anything. And all my slides from now on, they always have that little thing. And that's sort of a little trick for Zoom talks in the age of COVID. All right, so this talk is called Life Before Evolution, and obviously it's an origin of life talk. So the question really is, how can life originate if in fact you do not have the capacity of that evolution has, evolution that is this amazing process that can create all the complexity that we see. And now if you say, well, how can I even understand life before evolution, since some definitions of life actually use evolution as the primary qualifier of what life is. But obviously, this doesn't really work, for understanding the transition of life. So we may have to look at a different definition. And one that you might come across is this one by, or at least, you could look up that book, which is the famous book by Erwin Schrödinger called What is Life? Actually, if you take a look at that book, it actually has several parts. It has what is life, which is the physical aspect of the living cell, and has mind and matter in it, and even autobiographical sketches. This is a very famous book for a number of reasons. It inspired a bunch of physicists to start working on biology, in particular the people from the phage group, and Watson and Crick both said that they were very much inspired to work on the problem of DNA from that book. And the other one, I think, is that Schrödinger coined the idea of an aperiodic crystal, their DNA as an aperiodic crystal. But if you've ever read the book, you should also come to the conclusion that almost everything that he suggests in that book is in fact incorrect or wrong. So all the assumptions he makes about DNA are essentially wrong. All mind and matter is about the structure of the brain is also all wrong. So that can just remind you that even if you write a paper or a book that is almost completely wrong, it can still actually have great effect into the future. So if that's not how we define life, how should we define life? So I'll show you an alternative definition, which is life is a system that replicates the information about how to replicate that information. Now, some people have actually attributed that to me, and I've said that many times, but it turns out that John von Neumann actually made that point in 1948, before even the discovery of DNA. And so some people, like Sid Brenner, for example, have said that, if John von Neumann and Crick and Watson would have met, von Neumann essentially discovered, how information is stored, or the secret of life before Watson and Crick did. Because indeed, if you read his work on what he thought was life. It's very clear that he talks about information that is used in order to replicate the information. Now, fine, if you actually believe this, then you still have to ask, well, life is a system that replicates information, but what is information? And I sometimes give an entire talk about what is information. Here, I'm just going to show you a very simple way of understanding what it is without doing a lot of math. In fact, I barely do any math in this particular talk. So here's the colloquial definition, which is information is that which allows its holder to make predictions with accuracy better than chance. Right? So if you can make a prediction with accuracy better than chance, that means you actually have information about what it is that you are predicting. This is actually, of course, Claude Shannon's definition, and even though he writes it mathematically, it's a pretty good way to sort of colloquially summarize, namely that information is always going to be about predictions. If you don't know what the information helps you predict, it's actually not information. This is a common mistake where people say, well, if you just hand me something with symbols on it, it surely must be information. But no, if you don't know what you can predict, you have data, but you do not have information. Here's another example. Here, for example, suppose somebody hands you this piece of paper there with this sequence of letters. How much information is this? Well, you could ask, well, look, it's 15 letters, including spaces counted as a letter. Then, how unique is that sequence? Well, it's 1 out of 27 to the 15, 27 because 26 letters plus 1 space. That's 1 out of 3 times 10 to the 21. So if you want to know, what is that, how many bits it takes to specify that, it's actually 71.3 bits. Is that the amount of information? Well, I told you, if you don't know what this thing is predicting, then you don't know how much information it is. And predicting itself isn't really information. But, for example, you can imagine that this is actually somebody telling you who is the murderer in the game of Clue. I believe in Europe, it's called Cluedo. And now that we know what this piece of paper is actually trying to tell me, I can actually calculate the information constant. Namely, it is the logarithm of 6. It's basically specifying 1 out of 6 of these possible murderers. Okay, so in other words, it's not 71.3 bits, it's 2.6 bits. And clearly now, once you know what you can possibly predict, it's also clear, how much information is in there. So in other words, a sequence of letters isn't information until you're telling me what it is that you're going to try to predict with it. All right. Another thing I want to talk about with information, that it's actually fragile. If you take a piece of information and you are sort of allowing it to be exposed to the vagaries of an environment, and well, you actually if you're allowed to copy it all the time, like of course DNA is, it can be constant. Basically you can maintain information, right? But if you do not have a copying or maintenance process, then information over time will deteriorate. It is simply a consequence of the second law of thermodynamics. If you do not actively maintain, like via copying, information, it will decay. You could possibly, create a process where, the information is stored in a robust manner, but all it does is delay the decay. So in principle, and that's an important part, in order to make information stable, you need to have something like replication. But what could you possibly do, or how could a good amount of information, a merger, be maintained when copying is not available to you as a mechanism? You could essentially ask, well, some information can be formed spontaneously. Like, for example, here you see a spontaneous formation, but you also see that it will decay. Here's another formation, event, and decay, formation, event, decay, and so on. So we can ask the question, well, how high can these spontaneous information creation events go? For example, you could imagine that there is sort of a threshold up here somewhere where the information is large enough that you could actually have enough information to self-replicate. And so you could ask the question, could I ever have a spontaneous event that will sort of breach this, what I call the Darwinian barrier? So what is the probability distribution to find spontaneous information of size I? And if we had the relationship between this likelihood of emergence and information content, then we could answer that question. But what is that? Well, I actually will give it to you. It is a very simple expression. This likelihood is essentially exponential in the amount of information. So very small amount of information, high likelihood of spontaneous emergence, large amount of information I, extremely low spontaneous emergence. And in fact, if you turn this around, you see that the information content is really just minus the logarithm of its likelihood, which is, by the way, also how I calculated the information content for the Colonel Mustard case just earlier, but I just didn't show you where it came from. So if we have this formula, well, let's calculate a few examples. So for example, how likely is it to find the letter O? Well, first I have to answer, well, what's the alphabet? So like, okay, the 26 letters, lowercase a to z. Well, then if I assume that, in fact, these letters all occur with equal probability, then the answer is, well, 1/26, of course. The information content, this is the information actually to specify the letter O, would be 4.7 bits. Or here's another unit that you can use. If you take log base 26, then you can say that the information content of the letter O is 1 mer, where I can make polymers out of these letters, right? So how it is likely to find the dimer or? And the answer is, of course, the likelihood is 1/26 squared. So how likely is it to find the simple 7 mer origins? And the answer is, that's already 10 to the minus 10. See, this is the real problem of having this exponential dependence of the likelihood on the information content. And here you have one of these monkeys operating the typewriter, and you'll see that this monkey is going to have a very hard time to even write this simple sequence of letters. There's about 33 bits of information in the 7 mer origins. And again, what is this specifying? In this case, this word is, this sequence of letters specifies this particular word origins. So, but generally speaking, we will define the likelihood of a piece of information I as the number of sequences of a length n mers in this case, that carry the information I. So that could be if everything is in, if this is an n mer, and if I measure the information in terms of mer, then of course this I is going to be less or equal to n. And this is divided by the number of all of the sequences of length n. So it's like the density of information carrying molecules. That's this likelihood. So what do we find? So in order to find 5 bits of information, obviously to find it by chance is 1/32. To find 10 bits of information is about 1 in 1,000. And to find 100 bits of information is already smaller than 10 to the minus 30. Right? And so maybe you've heard about the infinite monkey theorem, which basically says, hey, if you have an infinite number of monkeys writing on typewriters, they will come up with Hamlet or any other a classic work of literature. Now let's check this a little bit. So let's take this phrase out of Hamlet and let's just make it a sequence of capital letters. What is the likelihood that our monkeys will actually create this sequence? And the answer is, well, that's a 30 mer or 141 bits. And the likelihood to create that is less than 10 to the minus 42. So forget about creating a Hamlet. Forget about even an entire army of monkeys trying to do that. They're not going to get even this phrase. So how many bits could conceivably arise spontaneously? Well, let's take a look at actually a self-replicator that was created in Jerry Joyce's group in San Diego. It's basically as a self-sustained RNA enzyme. That's actually a 43 nucleotides, so 86 bits. That has a spontaneous likelihood of 10 to the minus 26. which is extremely small because keep in mind the weight of the Earth in grams is 6 times 10 to the 27. You know, if you write these exponents, sometimes these numbers don't mean anything anymore until you compare it to a number that might mean something. So yeah, that thing is not going to arise spontaneously. Here's another example. The information content of a small viral protein, in this case, the HIV protease, turns out to be 75 mers, as I've calculated in that paper from a few years ago. The actual sequence is 99 mers, but only 75 mers of that are information. The likelihood of that arising spontaneously is about 2 times 10 to the minus 96. Now, think about how small that number is, given that there are only about 10 to the 80 atoms in the entire universe. So if these likelihoods were unavoidable, the likelihood of spontaneous emergence of sufficient information for self-replication is not just small, it is zero. It is zero not just on a finite Earth, it's actually zero in a finite universe. It cannot happen if these likelihoods were unavoidable. Now, what I'm going to show you is that, in fact, they are not unavoidable. Because there are assumptions underlying this theorem that I just showed you. That was namely that this likelihood is a number of sequences of length n that carry the information I divided by the number of all sequences of length n. This is correct if all of those sequences are formed with equal probability, which I'm sure you can figure is probably not likely. In fact, also if all the monomers in particular that make up any of these sequences are formed with equal probability. Like when I told you origins, O and R and I and G and I and so on, they're all formed with probability 1/26. Well, we know that in English, for example, these letters do not occur with equal probability. So what happens if, in fact, the formation rates are not equal? So again, the likelihood of the word origins, I told you, is about 10 to the minus 10. If I assume this uniform distribution for the likelihoods of any of these 26 different letters. So what I'm telling you is that the shape of this distribution is important. For example, if for example, one of these letters was missing, can never be formed, I, for example, here you see that it's still normalized. So this moved up to 1 in 25. Now the likelihood is actually 0 because you cannot make that word, right? In a random process. But now we can ask, well, first of all, what we did is, can we actually just test this? So Thomas Labar in my lab a couple of years ago, he searched 1 billion 7 mers, of letters for the words origins using this uniform distribution. And indeed, he didn't find any of them among these billion. And since the likelihood is actually 1 in 10 to the minus 10, 1 in 10 billion, that was actually expected. Now, suppose, in fact, I search 1 billion 7 mers for the word origins, but now using the distribution of letters that is found in the English language, which you find here. This is actually the probability distribution of English words. Okay, so for example, E is the most common. Next one is T, and so on. And that actually has, this distribution has an entropy of 0.89. Okay, the maximal entropy for the uniform distribution is 1. Okay, the more sort of pronounced these peaks are, the smaller this entropy. But the entropy of English is 0.89. That looks like it's pretty high, but in fact, you see that it's really a very uniform, a non-uniform distribution. And then we said, okay, if we search where we're creating E much more likely than we created J or an X or a Z, how often are we going to find the word origins? And in fact, he immediately found it twice. So that clearly means that searching with this biased distribution as opposed to a uniform distribution increases the likelihood of spontaneous emergence. So we call that the biased typewriter. So it's not the uniform typewriter of the monkeys, but now you have a typewriter that is biased, that it actually reproduces, the monomer, probability distribution of a functional molecule. In this case, functional just means English. Now, I wrote down the theory of what we actually expect for this likelihood, and I'm saving you all the mathematics of it. So what I found is approximately this likelihood is the dimension of the alphabet. Previously, I wrote it as an exponential, e to the minus, but in fact, here it's the dimension of the alphabet to the minus the, the information content, or in this case, the length of the sequence, but times the entropy that where you're drawing these monomers from. So basically, and we call this the abiotic entropy. It's basically the likelihood that the monomers are being formed in a spontaneous process. Like on Earth, for example, you will actually be able to form nucleotides spontaneously. You can form certain amino acids spontaneously. And certainly that doesn't have a uniform distribution. So basically that means that since this abiotic entropy is between 0 and 1 if it is a uniform distribution, and smaller if it is non-uniform, it actually reduces the sequence length that you need. And because it's in the exponent, that can actually have a pretty big effect. Let's test that, for example, with the Lincoln-Shostak replicator. So the likelihood of the Lincoln-Shostak replicator, if you don't have any biased search, is 4 to the minus 43 or 10 to the minus 26, as we previously mentioned. Now, if this entropy from the formation process, instead of being 1, is actually just 0.95, turns out that the likelihood is actually substantially larger than the unbiased one. If it is at 0.9, you get to 10 to the minus 23, and just having an entropy of 0.8 instead of 1 already gets you over 6 orders of magnitude increase in the spontaneous likelihood. In other words, it's a huge effect. Now, we can test whether this actually works for the spontaneous immersion using Avida, for example. In Avida, we have, of course, sequences of instructions that are executed on a virtual CPU and that have certain functionality. In particular, there's the possibility of having self-replication in them. So here's the instruction set, a typical instruction set. In fact, we did it in such a way that there's actually 26 instructions, so that we can have the lowercase English letters as placeholders for any of these instructions. There's a standard handwritten replicator that we sometimes use in experiments that has actually 15 instructions. And here it is written in terms of this alphabet. Okay. Now you can say, well, how likely is it? Well, this particular one, just by straight math, is extremely unlikely. It's about 6 times 10 to the minus 22. If you would be searching, by random, creation of any of these letters for this on even 1,000 CPUs, you would probably, I did the calculation, it would take about half a million years to find this particular one. It just shows you how rare individual strings are. Now, not all of these 15, monomers or letters in this are actually information. So in fact, the likelihood of that is much, much higher than 6 times 10 to the minus 22. How high? Well, we can actually search for this by using a uniform distribution to discover the information content. So how do we do that? Well, we actually created 1 billion random sequences of length 15 in Avida a few years ago. It was Thomas Labarre did that. And what he did is he found 58 unique self-replicators. That's actually quite a lot, given the fact that we thought that if all of these 15 MERS were information, it would take you 500,000 years. But he found 58. So from that formula, we can calculate the information content of these self-replicators, and that's just the number we found divided by the possible numbers that we tried. In this case, 10 to the 9. So that's about 5.6 MERS. Okay, so in other words, of these 15 MERS, maybe 5.6 or so are information. Maybe because, of course, it was not an exhaustive search. So we said, oh, could there be replicators of length 5? And the answer was no. We tested almost 12 million of them. Okay, but maybe 6. Now, of course, you could have said 5 is sort of weird because the information content is already more than 5. So we tested all the replicators of size 6, and the answer is no. We tested all 309 billion of them. So what about, we jump to 8, okay? So Thomas tested 1 billion sequences of length 8, and out of 209 billion, possible ones. And in fact, he found 6 replicators. Here they are. And you see actually, there's some very common motifs that you can find here. Here's a GB motif, here's the FG motif. So you realize that, in these self-replicators, they're not random sequences. There's syntax in it. Real motifs, just like you would find in maybe enzymes that have certain, replication motifs or sort of kinetic motifs. So now we can actually estimate the information content for the length 8 sequences. Remember, previously we estimated the information content of the length 15 replicators from the 58 that we found. And so here, in fact, we found something similar. We find that, in fact, this information content of these replicators is about 5.8 mers. So now the question is, can a biased typewriter help find replicators? Okay, because here we used unbiased typewriter, each of the 20 letters generated with equal probability. So what we did is we took these 58 n equal to 15 replicators that we found, and we create a usage distribution. How often do any of these instructions actually appear? So F and G, for example, were very common motifs. And then these letters V, W, and X are actually very important because they are responsible for dividing the code and from allocating the code. And so they, this line here is the uniform distribution. So you have a few that are much, much higher than uniform, and then a few that are actually much lower. So now the question is, if I use this distribution, this bias distribution for search, how many replicators do I find? By the way, it has an entropy of 0.91. And in fact, he found almost 15,000 out of 1 billion. And previously, of course, he found 58. So how does this compare to the theory that I wrote down? So the way we test that is we actually create a bias, an actual bias, as an abiotic bias, that is 1 minus this bias constant plus the bias constants times this entropy that we had found. So if the bias is 1, then you're searching with the actual distribution h of a. And if the bias is 0, you're searching with a uniform distribution. In other words, this bias is interpolating between the uniform distribution here and this highly peaked distribution h of a. And here you see, sorry, let me go back to what you see here. This is my theory, how the enhancement factor should increase with bias. And these dots here are actually the empirical enhancement. And in fact, at bias 1, here's the enhancement that we found, namely almost 15,000 over 6 that you found in the unbiased case. So you realize that biasing the distribution using that empirical functional distribution makes a huge, huge, huge difference. Now, the other thing we can ask is, if you find lots of replicators using a biased probability distribution, can you use the new set that you found to create a new distribution, maybe a more refined distribution, and use that to search for even more replicators? And the answer is yes, actually, that works quite well. So we did this actually for the distribution that we found for the L equal to 8. And there's only 6 of them, so that's a pretty coarse distribution. That's the distribution that we found from replicators of length 15. And this is actually a distribution for length 30, which actually, because there's so little information among a 30mer, that there's a lot of randomness in that sequence. So that's why you're seeing that many instructions actually occur close to the uniform distribution. But here you see actually, well, this shouldn't be here. But you see, in fact, that for each of these 3 different distributions, this is what you find in the number of self-replicators if you're using an unbiased distribution. But as you then rebias and rebias and rebias, then the number of self-replicators that you find increases quite substantially. This is an exponential or logarithmic scale. Okay, so in other words, if refining this distribution helps you tremendously in your your ability to find these very rare sequences in that space. So now we could ask the question, could a dynamical system somehow self-organize so as to rebias these monomer distributions? In other words, could a system sort of like bootstrap itself to search for into more and more concentrated areas of genetic space for these replicating molecules? So basically we would ask, is there some sort of process where you're starting out like this and you're going towards a more somewhat biased distribution to an even more biased distribution, and then maybe to a very highly biased distribution, which actually gives you an exponentially higher chance of spontaneous formation of information? And the answer is, we don't know. This would be understanding the origin of life, but nobody, as far as I know, actually can show you that there is such a path or how that would actually happen. So that is pretty much the way I understand the mystery of the origin of life, how you could come up with a process that creates this kind of changing of the probability distribution without actually having replication. If you do replication, of course, you're going to get very, 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 very specific distributions of that sort. So the road to self-replicators goes like this. So we know that if we have an unbiased process, then the only thing you can achieve are these small, spontaneous jumps, and they will decay. But you will not reach this Darwinian threshold. What you want is a process that somehow builds on itself, that you get spontaneous increases that get higher and higher and higher, because the monomers are being drawn from a more and more specific distribution that is actually biased towards functional molecules, up until the point where you get a jump that actually gets you over the Darwinian threshold, and then evolution can take off and create more and more information. So, but as I said, how we get from here to here, nobody knows. But we can also ask the question, well, how easy is it actually for evolution to take hold at that particular point when you have enough information? And so we can actually test this in IVIDA. We can ask, how likely is it, and what does this landscape of pre-Darwinian selection look like? So we can do that by actually creating the fitness landscape before evolution by measuring the replication rate of all the molecules in the landscape. So what you want to find out is, suppose this is all of genetic space. If you have a replicator here, obviously if there's a single replicator, there's no evolution. Okay, so in other words, if there's only one peak, that's it, no evolution. If there's two peaks, there still might not be any evolution, because these might be too far away from each other to be connected. Even if you have a number of self-replicators, the real question is whether, in fact, each of these self-replicators is surrounded by a cloud of mutated self-replicators where these somehow overlap, so that you can actually have a sequence that allows you to traverse this genetic space. Now, in AVIDA, because we can actually look at all sequences of small lengths, we can answer this question. So for length 8 replicators, we just have to, calculate or find the replicators and their replication rates of 209 billion of such things. And that was actually published about 4 years ago, and Nitash did this particular work, and I'll show you the results here. So he found 914 replicators out of these 209 billion. So now we can actually calculate the information content of these replicators exactly. It's actually almost exactly 5.9 mers. Okay. In fact, here's the entropy per sequence for those, and you see again these F and Gs, and you see also that each particular sequence absolutely has to have a V, W, and X. So each of the sequence, the likelihood of finding this one is 1. And the reason for that is that, V is the copy instruction, W is the allocate instruction, and X is the divide instruction. So of course, a replicator has to have that. And indeed, that's statistics of 914. So yes, indeed, all of them have that. But we also see, in fact, some very strong highly overrepresented motifs. FG and GB you've seen before, but there are others called the HC and the RC motif. In fact, one of the things we found is that there were really two very different types of replicators that we had in that landscape. One of them had these FG and GB motifs, and the other type had the HC and RC motifs. So their algorithm of replication was really dramatically different. Now, what I'm going to show you is a plot of actually all of the replicators, but sort of crunched together using what is called a multidimensional scaling. So here the axes are multidimensional scaling axes, which means that the distance here is actually proportional to the Hamming distance, the number of mutations it takes to get from one to the other. And a line is drawn if, in fact, I can have, these are mutationally connected. And in red, I have these FG replicators, and the blue is the other type, it's what we call the RC replicators. And in fact, there is never a line between the blue and the reds, even though they look, they're sometimes overlapping in the MDS coordinates. But in fact, they're not connected, these two groups. And what you find is that they form very, very tightly connected groups of sequences. Remember, there's no evolution that has taken place in this landscape. This is just the pure landscape. And it's very clear that a landscape like that is highly, highly traversable, even before evolution even starts to come into existence. Here is, in fact, its fitness landscape, whereas here's the MDS coordinate, the same one as I showed you before, and the replication rate. What you find, by the way, all of these are the red FG replicators. And down here, you see actually these other type of replicators, which have much, much lower fitness. And within this landscape, you see that a few of those have really high fitness, but they're actually surrounded by other replicators with also pretty high fitness. So these landscapes are actually quite correlated. They're not random at all, which makes them actually traversable. So what we find is that, for example, if we start 200 evolution experiments with all of these 914 ancestral replicators, we ask the question, which of those 914 is likely to be the ancestor of the final population after a good amount of time has passed? So after 50,000 generations in this case, because we want to see how common is it that one or another one of these sequences is mostly going to be the ancestor, or whether this is pretty much random. And the answer is actually it's quite how should I say it, non-random. So for example, these three sequences in oranges were the ancestor of the final population 130 times out of 200 combined. Whereas these green ones, all FG replicators, each of them was found to be the ancestor less than 6 times. And these gray ones never were the ancestor. So in other words, even though you have a landscape with 914 possible ancestors, if you throw them all together, most of the time it's one of those three that is going to be the founder of the population. In other words, evolution on this primordial landscape is actually fairly deterministic. Now you can ask, well, how does that scale up to bigger sequences, larger genetic spaces? So Nitash actually went through the trouble to replicate this entire experiment for the L = 9 landscape, just one instruction more. But one instruction more in this exponential space makes a big difference. Now there are 5.5 trillion sequences that you have to test. Now, if you test one sequence every microsecond, you still have to run this for 172 years. But fortunately, we have an HPC, and ICER has nicely allowed us to run this for a few weeks. And Nitash found actually 36,171 sequences. So we can now calculate the exact information content, and it turns out it's actually slightly less. The reasons why this information content actually depends on the embedding is a bit complicated and is described in that paper, and I won't have time to get into that. But we can actually understand, why embedding information into longer sequences, will change the information content. So what he found is that you actually have somewhat similar monomer proportions, but in fact the dimer proportions also change. So for example, you still see the HC motif being very prominent, but the RC motif that was over here pretty much disappeared. And so basically what we find is that in this very much larger landscape, you still have the same exact trends that you observed in the smaller landscape. And so we have confidence that if you go to much higher dimensional spaces and longer sequences, that we're going to see the same trends, that they were not just due to the small sequence. But it was also another thing that we realized, that we actually showed that if you look for sequences with a biased distribution, for example, one like that you are significantly, namely exponentially, reducing your search time. But you can even ask, well, what if I actually search with a biased dimer distribution? I create lots of FGs and lots of GBs and lots of HCs. What is the likelihood? I'm not giving you the information, right? You still have to make an informational molecule, but I'm creating small building blocks for you. It turns out that this is increasing the rate of finding functional molecules, again, by a very large factor. For example, if you would be looking for the words origins using OR, IG, and so on as the components, you will find that word, almost instantly. So in other words, another avenue towards the spontaneous emergence of life is, in fact, if some dimers are being formed, with high probability, and they are actually present in functional molecules. But that is still something that obviously we certainly haven't tried this search on actually nucleotide sequences, biasing them with the type of maybe or amino acid distributions that you find in functional proteins. But some experiments were done actually in Jeff Shostak's lab, which show that actually is a very effective way of finding functional proteins. So what we learned? We learned that life cannot emerge spontaneously without help, and the most obvious help is a bias in the monomer formation rates. In fact, in geochemistry, the rate of formation of monomers depends heavily on the local environment, like temperature, like salinity, and so on. On an early Earth, you can imagine that there might be millions of different geochemistries or geochemistries with different salinities and different elemental compositions, each with their own entropy distribution. And maybe one of them is just right to bias the spontaneous emergence. And you really need only one, right? Now, if there was a guided self-organized process, that would help lead you towards that probability distribution, even better, but we don't have evidence for such a process as of yet. And finally, once the Darwinian information threshold is breached, it's very likely that evolution will take hold almost immediately, because what we found is that space seems to be traversable. Now, another thing we learned, or at least that I learned, is that the concept of information really is key to understand life in the universe, and certainly the spontaneous generation of life. I believe that all life anywhere needs to be encoded in information molecules, whatever alphabet they might use. This first information may be extremely rare, but once it's there, probably Darwinian evolution can help create more complexity from it. It needs environments where monomers are produced at rates close to how they would appear in replicators, and those would be highly favored to be the cradle of life. And that's pretty much all the lessons I wanted to get across today. So let me thank the people involved that helped me with this work. Foremost, Nitash and Thomas, who both were the lead in the two different projects that I showed you, and Arend Hinze also helped us in the early days of this project. And at that point, I'm happy to take questions.
Thank you for that great talk. We can open it up for questions. Please feel free to unmute yourself and ask. And if we get a lot of questions at once, the raise hand thing is now under the reactions button. So if you raise your hand, I'll know you're waiting. You can also use the chat if you prefer.
Right. So Joe Graves, for example, comments that the infinite monkey theorem actually claims that you could create the complete works of Shakespeare. That's correct, or any of that. But they never mention or do the calculation that, you know, in a finite universe, this is so far away from the possible that it's even laughable.
Chris,
Yes.
on the missing ingredient that would be able to dynamically create these conditions, what about self-catalytic cycles, where certain combinations of molecules increase their own probability, and then you start changing the distribution of MERS
Yeah.
in the...
Yeah, so Anselmo is mentioning, you know, a possible route to the origin of life that in fact was theorized already quite a while ago by Manfred Eigen and Peter Schuster, who thought that if you had some molecules that catalyze the formation of other molecules in a closed set, that might actually have these self-organizing processes that could create, you know, a drive towards higher complexity. Unfortunately, nobody has ever actually been able to demonstrate this. So the only thing that people have been able to show is that you could actually have such, you know, stable catalytic cycles, but also nobody's actually been able to make them spontaneously emerge. Or in other words, these molecules already carry enough information to be, in a sense, self-replicating, even though they're not really replicating each molecule, but the group, you know, is replicating. And we could calculate the information content of that group. And the answer is it's post-Darwinian threshold already. So the question is whether you could have small molecules, you know, affecting each other rather than catalyzing their replication, you know, are just doing some sort of catalytic reaction that leads toward information maintenance. And nobody knows what that could be. I mean, I have thought about this for quite a bit, and there are ideas that where the idea is that you can have a passive template-based replication due to clays, for example. In other words, the molecule does not encode the replication; it's the substrate that makes replication possible. You know, it's actually not that far-fetched. In clays, you can actually have template-based replication, but this is not life because the information to replicate is in the substrate, it's in the clay, it's not in the sequence. What needs to happen via this self-organized process, that the information how to replicate needs to seep from the environment, from the clay or whatever helps it, into the sequence. And there are ways you can think that that might be possible, but how that actually happens, nobody knows. But good question. I'm sure there must be more questions. They don't have to all be as good as Anselmo's question.
I'll throw in a question. So I'm David Baum, and Joe sent me the link. I hope it's okay that I gate crashed. So I'm at the University of Wisconsin. But anyway, I just want to, I really enjoyed the talk. But of course, you know, you were aware of this, but of course, if the word you needed to self-replicate was a word like jazz or something that was anti the bias, you're making your life harder. So I do see this issue that you raised of the bootstrapping or ratcheting yourself to the right bias seems absolutely critical for this to solve.
That's right, and you're absolutely right, and that's also again a very important insight. I didn't have time to talk about it because Thomas actually did such an experiment. So, for example, we figured that if there is a sequence that is being produced by another sequence, not replicated but produced, that sort of would be broken down and be put back into the environment, so that, you know, the environment would be biased by the monomers that, you know, that have been, you know, produced. And the answer is that's terrible, because you're actually moving away, because if there is a single molecule that's necessary and it's not being produced at the beginning, well, it's never going to be introduced into the soup. And so that process, and Thomas showed that in Avida, actually leads you away from where you want to be. So it needs to be a pretty special a kind of bias. And you know, if it was obvious, somebody would have found it already. We really don't know what it is. Maybe there is something special about biochemistry that allows you to do that. Maybe there's something special to the kind of, you know, ribose-based nucleotides We don't know. We don't even know whether the first alphabet was, RNA types, or whether in fact it is ribose based. I believe it's probably based on ribose, but maybe not the kind of nucleotide that we are used to. It's a big mystery. I mean, that chemistry, primordial chemistry, is most likely completely lost to us.
I will say that we do have a model that I think does does ratcheting up, but another time, if you're interested, I'll tell you.
Okay. Whoever finds this, is going to be looking at being quite famous, because it is one of the biggest questions that science, is still trying to answer.
Yeah, I'm not looking for that, but if you're interested sometime, I can tell you about it.
Yeah, I'd be interested in seeing that. Yes, I'm sure you can find my email. It's just adami@msu.edu. Anybody else?
I don't have a question. Chris, do you mind if I plug my talk for next week?
Oh, please. That's a good idea.
So if anyone found this interesting, I'm going to be presenting next week, also an information theory-based talk, and I'm going to be looking at how we can use information to predict data flow through brains. That's all.
Yeah, so that's going to be interesting, because we just got a preview of it in the group meeting a couple of hours earlier. So
Well, just for clarification, Cliff's talk is actually in 2 weeks. We're doing
2 weeks.
seminars every other week, so that'll be February 12th.
Oh, I have more data then. Great, thank you.
Excellent.
For making sure that people don't show up with nobody there.
So Chris, this is Bob Root Bernstein. I'm in physiology. I've been working on this problem of the pre-life issue for probably 25 years now or longer. And we do it both theoretically and experimentally. There are a series of both theoretical papers and experimental papers that I will send you. Basically, what they show is that there is a very strong biasing mechanism which involves molecular complementarity between small molecules. And what this does is the molecules survive against thermal degradation, things like that. So you get some molecules that survive at higher rates than others. They can catalyze each other's reactions so that, for example, we've actually done this in the lab Miller did way back in 1953 and afterwards. You can make amino acids with, just pure gases. And what we've been doing is running this thing over and over again, and you start getting dipeptides, tripeptides, et cetera, spontaneously within days. So we can start making very biased sets of chemicals and show that we get fairly high yields up to millimolar very quickly in these kinds of systems.
Yeah, and I believe that really must be the avenue. Like I said, when I thought about how to get information from a catalytic surface into the sequence that is being template directly, so at first you have a random sequence, no information in it, but the kind of processes that you just mentioned, like for example, having a sequence that is easier to copy, right? Because of the molecular composition, and that creates non-randomness. That already is information, because now that these things are being copied more quickly, they will increase in abundance. And if there are catalytic elements, just a few monomers forming dipeptides and so on, they create a non-random sequence, and then information seeps from the substrate into the sequence. And if one can show that can actually go on and on and on and on until reaching the Darwinian threshold, then you have a winner. But I'm absolutely convinced that the kind of mechanisms you just described are indeed key to try to understand this biased accumulation. Yeah,
so there's also a bunch of theories which I think complement yours that actually come out of economics, which have to do with what a system is. And basically, if ways of eliminating all the possible probabilities in a system to make it much more likely. So I'm just going to send you the papers. I don't want to try to explain this all right now.
Super.
It's a talk in itself.
Very-
Anyway,
Yeah.
I hope we have some more discussions. I really want to hear what you have to say about, the approach we're taking. I think it complements exactly the problems you're asking about.
Great, I appreciate that. Go check the chat window. Okay, all right. I think we're out of questions. So let me thank everybody again for dropping in. Having some fun old times, and I guess we'll all hopefully see each other again 2 weeks from now for Cliff's talk.
Thank you, Chris.
So,
Thanks, Chris.
Good to
thank
see
you.
everyone.